Tonight's presentation, Counting Seabirds in Puget Sound, How Difficult Can Be. Our speakers are Matt Farr and Toby Ross. Matt is a postdoctoral scholar at UW's Washington Cooperative Fish and Wildlife Research Unit at the School of Aquatic and Fishery Sciences. His research has focused on population and community ecology with an emphasis on modeling wildlife abundance and dynamics. As you will hear in more depth very soon, Matt applies quantitative methods to evaluate strategies for monitoring marine birds in order to estimate bird populations, their status, and trends. In his research, Dr. Farr interacts with U.S. Fish and Wildlife Service, the Washington Department of Fish and Wildlife, and other entities. Toby Ross has been a dedicated global conservation person in efforts for over 30 years. He was science manager at Seattle Audubon, now Birds Connect Seattle, for 8 years, where among other duties, he organized and managed community science programs. One such is the Puget Sound Seabird Survey, launched in 2007. This year, he is responsible for training and coordinating over 270 PSSS volunteers distributed across nearly 160 sites around the South Salish Sea. Toby also runs Alight Tours, which offers guided birding adventures worldwide with a focus on conservation and community engagement. Gentlemen, we welcome you this evening and thank you for the effort you put into this presentation, and we look forward to hearing from you.
Okay. So, hello everyone. My name is Toby, and tonight I'll be presenting an overview of the Puget Sound Seabird Survey. As a lot of you were coming on the screen tonight, I saw a lot of familiar names from past and present. So thank you, as always, for those who have taken part over the years. Many of you are going to know what I'm going to say tonight, but maybe you'll pick up a few things here and there. So let's get going. So the Puget Sound Seabird Survey, or PSSS, is a community science volunteer-powered program that monitors marine waterfowl throughout the South Salish Sea. The program was created by Seattle Audubon, which is now Birds Connect Seattle, to monitor seabirds in response to a number of perceived declines in seabirds. With Birds Connect Seattle's history in community science, we knew volunteers would be a great way to help us with this work. The then Seattle Audubon Science Committee designed the program in 2007, and then we piloted the program over the winter of 2007, 2008. And the program continued for 14 years while at Birds Connect Seattle. In 2022, Birds Connect Seattle decided that they could no longer manage the project, and so PSBO stepped in and adopted the program where it continues today. So why Puget Sound? So it's the second largest marine estuary in the US after Chesapeake Bay, and covers nearly 3,000 square miles. It supports significant populations of wildlife, including about 200 species of fish, 100 species of waterfowl, and 13 types of marine mammal, including, of course, our magnificent orca that we get to share. The region also has important cultural significance, economic benefits, and is home to about 4.5 million people. So it's really vital that we keep it clean and healthy. So why seabirds? Well, they make great indicators of marine ecosystem health. They're directly linked to the marine environment through food, roosting, and breeding. They're easy to see from land and are relatively numerous, whereas most other marine organisms are hidden underwater. And they're generally easy to ID and count, which Matt will talk a little bit more about a little later. So there are many threats to seabirds. Some of the key players are climate change. I know that's nothing new for anyone, but climate change, especially marine heat waves, which affects food availability, which impacts breeding success and mortality. Some of you may have heard about the incident in 2016 when hundreds of rhino auklets washed up in the Strait of Juan de Fuca, This was due to a marine heat wave, aka the blob, resulting in disease and lower reproductive rates. Thankfully, burrow occupancy and breeding metrics have returned to pre-blob levels now. Habitat loss, another classic. For example, loss of old-growth forests leading to marble mulet decline due to lack of nesting habitat. Again, many of you know, I'm sure, that marble mulets rely on old-growth forests for nesting, like this image here. And shoreline armoring and modification, which results in loss of nearshore foraging habitats. And of course, there are a bunch of other threats as well, but I'm keeping it just to those three for tonight. So the survey protocol. Surveys are point count surveys, all conducted on land at predetermined locations. One survey is conducted per month, October through April, and this captures the movement of these birds, mostly migratory birds, as they enter and as they leave our region. And of course, the months in between when they're overwintering here. All birds sitting on the water are counted within 300 meters for a maximum of 30 minutes. Surveys are conducted at every site at nearly the same time, synchronized with high tide to ensure the maximum amount of water in front of the observers. All surveys must be conducted within a 4-hour window, which is 2 hours either side of the high tide. And yes, every site, 170 odd sites, are all surveyed relatively within the same 4-hour period. So what are we counting? We're counting all the waterfowl that depend on the marine environment, which over the course of this program have been about 70 species, 70. Including alcids, gulls, sea ducks, geese, loons, any bird that rests on our marine salty water. Seabirds have different diets, of course, and so gathering this PSSS data also tells us what's happening with their food as well. Here are the 4 sort of main groups: piscivores or piscivores, of course, fish eaters, for example, rhino auklets and marble mulets. We have herbivores, of course, plant eaters, cackling goose and brants as examples. Benthivores, so food living on the sea floor. Examples here, pigeon guillemot and surfscoda. And omnivores that eat everything, horned grebe and Barrow's goldeneye. So site selection. All sites are located on publicly accessible shoreline, including man-made structures like docks and piers, things like that. We have data collected from 189 locations around the region, but currently have 165 active sites. And I've got a map on the next slide to show you those. The only watersheds without survey sites are Hood Canal and Skokomish. And we're just about to finish up our 17th season of data collection. So here's a map of all of our survey sites. All the little dots you'll see are survey locations. The different colors denote the different counties that they're in. You can see all the counties on the left-hand side. As you can see, we go all the way up to the Canadian border, up to Blaine, all the way down to Olympia, and all the way out west to Near Bay, and everywhere in between. Yeah, so all 11 counties that have marine shoreline have survey sites currently, and 14 of the 16 watersheds that lead into the Southern Salish Sea. The 2 missing are Hood Canal and Skokomish, and those don't have any sites just because of capacity. We've used various funding apparatus over the years to sort of help us expand, and we just haven't expanded to there yet. So if anyone's in the audience who lives in that area, who is interested and willing to take part in the program, let's chat, and maybe we can set up a survey site near you. So this program relies almost 100% on volunteers, community science program. It will be impossible without all of these folks. I work half-time, three-quarters time on this project, and I could not do surveys at all of those locations every month for 7 months. Impossible. So we rely entirely on volunteers, and over the lifetime of the project, we've engaged over 900 people, which again, extremely thankful to all of them. And here are some of the faces of the people who have been taking part over the years. Some of you may recognize yourselves. I see Elaine in there, and who else is online tonight? So yes, those are some of the people who help us out. Of course, not all 900, but a selection. Each season, we engage, although we've engaged 900 over the lifetime of the project, each season we need about 270 volunteers to make the program run. And I aim to create teams of about 4 people. The more, having 4 people gives more pairs of eyes to improve accuracy of counting, shared knowledge of the protocol, survey protocol, and also species identification, of course, and tracking which birds you've counted and which birds you haven't. So teams, I always try and team people up. Every volunteer takes their Seabird ID quiz, which helps me create balanced teams, try to make sure that every team has somebody who's fairly confident with their Seabird identification. And of course, that helps ensure that the data is pretty decent. And then other members of the team can perform other activities like filling out the data sheet and spotting and things like that. Training sessions are held in September each year at different locations around the region, and data are submitted to a dedicated website, and that data is validated each year for accuracy, make sure everything is there that needs to be there and any issues can be cleaned up. Applications of the data. A paper was published in 2015 that identified areas where certain species congregate, which we termed hotspots, and where they do not congregate, which we termed cold spots. You can see in the map there with the dots, the red and orange dots are where pigeon guillemots, for example, in this example, they tend to congregate in those areas, and where it's blue, they tend to not congregate. We covered about, I think, 15 other species in this paper, and it's going to be available on, I think, one of the links. And then oil spill data. We get all of our volunteers to gather oil spill data on a monthly basis. When they do their surveys, they are looking for buffleheads, specifically male buffleheads. As many of you know, the plumage that is at the waterline is bright white, and so if they ever come into contact with any oil on the water, that plumage will soak up some of that oil and end up looking sort of muddy along that waterline. So if any of our volunteers, and often that will denote that there's been some oil in the water, or that bird has come into contact with oil. So yeah, folks record that information on the data sheet, and that gets sent back to me. And if we do find a significant number, that information gets sent to the authorities. We also have trained volunteers that can help in the event of a mass oil spill. Because our volunteers live all across the region, we can get eyes on the water really quickly in the event of a spill, and can identify seabirds that have been affected by the spill. And of course, they can get eyes on areas that have not got oil, which is also equally important. Current research, super excited that we've got 2 projects right now. Dr. Matt Farr, the University of Washington, who we'll be hearing from momentarily about his work. And then we have another project working with Birds Canada. Birds Canada is Canada's only national organization that is dedicated to bird conservation. They're going to be combining Puget Sound Seabird data with a project that they run, which is called the British Columbia Coastal Waterbird Survey. It's like a sister project. Our project was based on their project. Their project's been running a little longer than ours. And so you can see we're combining this data in the map there. Their survey locations are in the red dots and ours, of course, the blue. So they'll be combining those data sets to examine trends across the entire Salish Sea. And we'll make sure that the results are openly accessible. We'll be adding our data and results to their Nature Counts platform, which is what Birds Canada manage, which will enable resource managers to incorporate the findings from this project into their management actions, which is, of course, what we want from this kind of work. Both these projects will provide us with truly the first abundance estimates and trends on how the various populations are doing. We've been collecting this data for all these years. We haven't been able to do a ton of significant, analysis with it. So we're finally doing that now. And I am super excited about these two projects, and we'll be sure to share the results with everyone once they're complete, whether through WASP presentation or something. Hopefully we'll be invited back to do that at some point. And that's going to be all from me tonight. Please volunteer if any of you out there who are PSSS curious. Obviously get in touch with me. There's my email address, or visit the Puget Sound Bird Observatory website. The link's there, where you can use your phone and the camera on your phone to put it on that QR code. Of course, we also do need folks. If anyone's living in and around Olympia or the San Juan Islands, or people willing to venture west of Port Townsend, we need you. It's been a little tricky getting folks up in the islands last season, and so yeah, I encourage any of you who are interested to get in touch with me, please. And of course, we want to appreciate our funding partners, Washington Sea Grant, CREO, which is who Elaine was mentioning earlier, Conservation Research and Educational Opportunities International, Sustainable Path Foundation, and of course, Puget Sound Bird Observatory community. I know Elaine's going to run ask for some questions later on. So if there's anything, please note it down for now, and we'll hopefully answer them a little later on. Okay, well, thank you. I'm going to hand over and stop sharing and hand it over to Matt. Okay, Matt, over to you. Great.
All right, thank you, Toby, and thank you everyone for joining me this evening, and thanks for the Society for having me, and I'm very excited to share some of my research that I'm conducting at the University of Washington on seabirds. And specifically, I'm excited to talk to you all about how we estimate population size for seabird species, and why it might be more difficult than you think, and definitely more difficult than just using your fingers to count. So that being said, my job at the University of Washington is essentially a glorified counter. I'm a postdoctoral scholar, and my research is at the intersection of statistical modeling and wildlife ecology. I personally worked on a multitude of species, from monarch butterflies to spotted hyenas, but at UW, my focus has been primarily on seabirds and on sea ducks. And so here's a photo of me counting one white-winged scoter. And then the other photo is of me holding a pigeon guillemot. And that is work that my lab is doing out on Protection Island, doing banding and nest box surveys. I do get to go out in the field occasionally. And I've actually participated, this is my first year participating in the Puget Sound Seabird Survey. So I do get out sometimes, but most of my work is on the back end, where I'm using statistical modeling and programming languages to kind of understand the trends and the distribution of birds and other wildlife species. So before I jump into the specific project I'm going to talk about this evening, I just wanted to highlight some of the other work that my lab is doing. So I'm part of the Quantitative Conservation Lab at the University of Washington, and we're within the School of Aquatic and Fishery Sciences. The principal investigator for this lab is Dr. Sarah Converse, and our lab is somewhat unique because Sarah is actually a federal employee. She works for the USGS, and we are part of the Cooperative program for the USGS. And it allows us to have sort of a unique place between academia and sort of the federal and state level agencies where we get to do a lot of applied research. And in addition to working in Washington State, our lab works throughout the Pacific, including Channel Islands in California and all the way over in French Polynesia. But today I'm going to be talking about a project that we got funding last year through the Washington Sea Grant in collaboration with Toby and the Puget Sound Bird Observatory. And essentially what we're doing is trying to combine some agency data from the Washington Department of Fish and Wildlife with the Puget Sound Seabird Survey. And the idea there is to put these two data sources together, and it'll help us understand sort of the long-term shifts in marine bird use in Puget Sound, and also understand some sort of seasonality and changes that we're seeing during the wintering portion of their annual cycle. And so when you are joining different data programs and sources together, you get the opportunity to work with a lot of great people. And so I just wanted to acknowledge some of the folks that I get to work with, including Toby and Puget Sound Bird Observatory, also the Department of Fish and Wildlife, and a bunch of other agencies that either are helping us or provided some funding. In addition to that, I'd also like to acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. Okay, so I'm sure a lot of you are, very familiar with what seabirds and sea ducks are, but I just want to highlight, why, we're focusing on these sort of species. And one of the important parts is that these species really rely on marine ecosystems for at least a portion of their annual cycle, if not the entire year. So just to give an example, here are 3 different seabird species. All of these species can be found year-round in the Salish Sea and in the Puget Sound. So we have the marbled murrelet, the rhinoceros auklet, and the pigeon guillemot. And then somewhat different are sea ducks, 3 different scoter species, and they're migratory species, and so they winter here in the Salish Sea, but during the breeding season, they're actually migrating north to northern Canada and Alaska, where they spend the breeding portion of their annual cycle up north, and then they return the following winter to come down to the Salish Sea for that overwintering period. And there's actually 11 different sea duck species that exist in Washington. And again, these species are using the marine ecosystem at different portions of their annual cycle. And part of that can be during the breeding season. So there's a lot of different breeding ecology for seabird species, like tufted puffins or colonial species that use burrows. And so they're relying during the breeding season on this marine ecosystem, right? But then you have something like a white-winged scoter that's migrating up to Canada and Alaska, and it's actually nesting inland on these inland bodies of water and relying on aquatic insects and plants during the breeding season. So they're not necessarily using the marine ecosystem at that part of their annual cycle, but then they migrate back down and are in the marine ecosystem during the winter time. And then there's something like the marbled murrelet, which is in Washington. It uses the marine ecosystem, but then it breeds in old growth forest. And so even though it's not necessarily going all the way up north to Canada and Alaska, it still has parts where it's using the marine ecosystem, and then maybe parts that it's not. And then a lot of these species do forage or have diets that are marine related. Here we can see a picture of a tufted puffin foraging for some small fish, and then also a surf scoter who looks like they're on the verge of eating a crab. And so their diets can include fish, fish eggs, crustacean, bivalves, algae, aquatic insects, and plants. And so the Salish Sea marine ecosystem is important for a lot of these seabird species. And the habitat that it provides is very critical toward them, if it's not the full year, at least part of their annual cycle. But over time, we've seen changes in this ecosystem, and we're starting to see maybe potential decline in some of these species. And so we're all very familiar with the metropolises of Seattle and Vancouver, which, you know, span from the southern Puget Sound into the Strait of Georgia. And the population has increased rapidly. At the turn of the century, there was maybe around 100,000 people. And, you know, across the entirety of the Salish Sea, it's projected to reach 9 million this year. So that's like a 100-fold increase in little over a century. And so with that, we've seen different disturbances within the Salish Sea. We've seen things like resource extraction from commercial fisheries, aquaculture, forestry practice. We've also seen land conversion like urbanization, farming, again, commercial forestry, and different sources of pollutants like oil spills, like Toby had mentioned, other sort of point source pollutants, and then also, maybe runoff from animals as well. And so in addition to these sort of human disturbances, we've also have these sort of large changes, but also that have local impact as well, from sort of climate change and changing ocean conditions. So here we have two different large-scale ocean oscillations, and these sort of changing how the oscillations have changed over time has been linked with things like warming ocean temperatures, rising sea levels, changes to water chemistry, and even ocean acidification. And so for marine birds, what this looks like is potential sort of what we call bottom-up effects, where they might see changes in their sort of prey availability or in their distribution, which can impact, you know, the survival of these birds. And so to combat these changes, the Puget Sound Partnership was formed, and it has a mission to sort of recover and conserve the marine ecosystem within the Puget Sound. And multiple goals exist for this sort of recovery plan. But what my work really focus on is this goal of a thriving species food web. There we go. And so within this sort of overall goal of a thriving species and food web, we have these sort of different vital signs. And again, the one that I'm focusing on for my research is this vital sign of birds. And so what a vital sign does is it really gives us an idea, or it indicates if this marine ecosystem is healthy or not. And we can measure sort of how well this vital sign is doing through different indicators. And so for the bird vital sign, we have indicators for both terrestrial and marine birds. And specifically, it's the abundance and trends of these bird populations. And so here are the different indicators. There's 4 indicators, which is a group of 6 different species. There's the marbled murrelet, the rhinoceros auklet, there's the 3 scoter species, and then there's also the pigeon guillemot. And so we can use these indicators to give us an idea of how the overall marine bird population is doing in the Puget Sound. And then that can give us an idea of how this vital sign, which represents the overall ecosystem health of this marine area, how well it's doing. And we can see in just some of the data that's presented here, the marbled murrelet is declining by around 5% per year, and where the scoter species in combination are declining roughly around 2% per year, with the data shown here, where the rhinoceros auklet and pigeon guillemot are seemingly doing a little bit better and have more stable populations. So in addition to sort of this recovery goals, DFW is also tasked with setting harvest quota for sea duck species. And so sea duck species are game species that can be harvested by hunters. And so the department needs to get pretty reliable information on the population abundance and the trends for the sea duck species to be able to set those harvest quotas. But due to sort of some of the ecology of sea ducks and the phenology of when they're, arriving here during the winter time, makes it somewhat challenging to get that information through different data collection methods that are maybe more typical. And so I want to just kind of highlight what I mean when I'm talking about abundance and species trends. And so I kind of want to define what those are and how I'm thinking about it. So let's assume that we're interested in surf scoters. We want to know how many surf scoters are in the Salish Sea. And so we can, the abundance is essentially the population size or the total number of individuals, right? And so that's sort of what the definition of abundance is. It's just total number of individuals in a area. And then we can think about how this population changes over time. And so we can see here this sort of delta N represents population change, right? And we can break this up into 4 different processes or demographic rates, right? We can have individuals that are born into a population. We can have individuals that immigrate into the population. But then we can also have a reduction from either deaths, so individuals that die off, and then also from individuals that immigrate out of the population, right? And so we can then collect data on these different demographic rates, or we can actually just collect data also on the overall size of these populations. And then this can help us estimate, what's going on as far as the status of these birds and the trends and their trajectory. And so there's a lot of different monitoring protocols in the Salish Sea and Puget Sound that exist to collect information on seabirds and sea ducks. There's boat-based surveys, there's colony surveys, there's banding surveys, and I don't do all of these. They're just photos of me by chance. And then there's also aerial surveys. So I'm going to talk a little bit about the Washington Department of Fish and Wildlife's aerial surveys, because that's part of the data that we're going to be using for this project. And so the department has been flying these aerial surveys in the wintertime around December, January, since 1994 up till now. And they fly these flight patterns, which is this sort of red little transects that you can see here. And they fly it once per year. And during these flights, they have 2 observers on either side of a plane who are counting the number of birds they see and then identifying each of those birds to species. And so people who are familiar with these aerial survey data know that they have a lot of issues that can kind of compromise the quality of your estimates if you don't account for them. But those who are not sort of familiar with these concepts, I'm going to do a couple of what I like to call like quizzes or interactive sort of game where I'm going to be showing some photos of different images from an aerial survey. And you guys can, as I move through these images, you can try to count yourself how many birds you see and try to ID them, the species that you see. So we have, I think, a couple of these to go through, and just this is just to kind of give you guys an idea of, maybe the issues with this data. Okay, so this is our first image. We're flying along, and we see this flock up, and we've passed the flock, and we've already needed to count the number of birds and ID the species. So I'll just give you a second to think. And that species there was brant, and then the total count for that species was 106 birds. All right, we're going to move on to the next one. Okay, it goes quick. We're moving quite fast, so you have to be pretty good at doing these counts. All right, so the species ID for that one, we just had scoter species, right? We didn't even get down to the individual species, maybe too challenging. And the count for that was 522. All right, here's the next one. All right, maybe this one might be a little bit easier as far as the ID, better quality photo. So that one was Canada goose, and there was 510. All right, we have two more. This one's a little different. It's over land. The birds are white, so maybe a little bit easier on the species ID. So that one was snow goose, and the count quite large, 2,960. And then here is the last one. Okay. Oh, again, Scoter species, and the count was 722. So I'm hoping from those different images that you can see that getting the IDs is tough, but getting the counts is even harder. And there's a lot of inaccuracy in humans' ability to count something that large that quick. So here's sort of a figure that kind of shows that. So there was a study that actually sent out something very similar to what I just showed you to people who do aerial surveys for different agencies. And they gave them these quizzes, and then they sort of measured the error between the real flock size versus what the quiz respondents were saying. And we see this pattern where as flock sizes increases, the amount of error that we're getting in our estimates is much higher. And then this is showing sort of a comparison between what the quiz respondents were saying versus what the true flock size was actually at. And so that blue line, if you're on that blue line, you did perfect. You said exactly how many birds were there in the image. And then the orange dots are representing the average quiz respondents' answer. And we can see this pattern forms that we start to have a dip in what respondents are saying. And so humans just have this tendency to always underestimate the actual flock size. And so there's sort of ways people go about trying to quickly identify the size of a flock. And I do this myself when I'm out trying to count the number of birds, is you look at a group of birds, you count maybe up to 10 or 20 or something that you're comfortable with, and then you start to extrapolate that sort of count across the entire flock, right? And so the top one, that they just count to 10, and then they start moving that sort of area of 10 to the entire flock. And down below, it's all the way up to 100. But an issue that exists here is demonstrated in this photo on the right. We might count that first one, circle, expand it to the rest, but the issue is that the density of the flock isn't consistent across it. And so there's actually a higher density in the flock at the back. And so if you took those 25 birds and extrapolated it out, you would actually still, again, be undercounting the total number of birds there. So it's not a perfect method, but it is a way to quickly try to estimate how many birds are there. And then obviously there's the issue of misidentification. I think a lot of people who participate in citizen science or recreational birders are very familiar with misidentification because it's usually what people hone in on as trying to minimize when they're out collecting data or just birding recreationally. And so here's just an example of two different golden-eye species that we have and that look pretty similar. So these are kind of the two big issues that exist in sort of this aerial survey data, is this idea of misidentification and this other concept of over or undercounting, which we call imperfect detection, or not being able to identify the total number of birds that are there and missing some individuals. So here are a few resources. I believe Elaine sent this out in an email, but I'll just leave this up for a second. Essentially, these are different sort of guides or resources that folks can use to help them improve their ability to count large flocks. So specific to getting better at counting flocks. And this isn't just, for aerial surveys. If you participate in the Puget Sound Seabird Survey, they can be very large flocks in those surveys as well. And so it's a similar sort of concept when you're trying to enumerate how many birds are there. So Washington Department of Fish and Wildlife were really familiar with this issue. And so they ran this camera survey in 2012 to be able to see how many birds are actually there. And I'm sure a lot of folks are like, oh yeah, why don't you put cameras on the plane? And so they did do that for a single year. And we built an analysis to sort of compare what the cameras were saying to what the observers were saying. And what that basically looked like was we had this data coming in from the camera. We also had observers up in the plane who were recording the number of birds and the IDs of those birds. And then we could compare sort of the compositions of both of those data. So the total number of birds and the species that were being seen and the numbers with those corresponding species, and we could develop these correction factors. And the goal was then to take these correction factors, use them in this long-term survey data to sort of correct for the fact that we're mis-IDing birds, we're undercounting the number of birds. And so this can kind of improve our abundance estimates and prevent us from inaccurately, making some inference on our estimates from our model. Okay. So this project, one of our very first goals was to get accurate estimates of abundance and population growth between years. And we kind of used that auxiliary study in conjunction with the long-term data to do that. And then we also wanted to understand what are the drivers of these trends and the distribution in the winter for these different marine bird species. And so we're kind of in the process of doing that now. And so we have some things that we think might be changing the trend, our population growth. And so those are sort of those demographic rates that I talked about earlier. And so one thing that we're interested in is sort of capturing maybe some differences in growth across the Salish Sea, using different sea basins to capture that variation. And then we also have sort of these large ocean oscillations that may be driving bottom-up processes in sort of changes in prey maybe over time. And then at the same time, we're also interested in where the birds are when they're overwintering, right? And can we sort of be able to find the relationship through some of these environmental variation in the Salish Sea and tie those with where the birds are. And so depth is a very depth has a strong relationship with sea ducks and many seabirds where these individuals are foraging at certain depths. And a lot of them are diving down to get their food. And so we included that, and then maybe sea surface temperature, trying to explain maybe where there's differences in prey as well. And so we have some initial sort of results from this work just using the aerial surveys, where we can see here, this is the community-wide predicted abundance. And so we've predicted from where we've surveyed on those transit routes out to the rest of the US side of the Salish Sea. And we can see this is just for sea ducks. So those 11 species of sea ducks, we kind of see this slight increasing trend, but really pretty stable, which is, somewhat surprising considering some of those scoter species looked like they were declining in those former images. And we're also able to get estimates of, mean population growth. And so the idea here, it's a multiplicative relationship. So if you're at 1, where that red line is, you take your abundance from last year, you multiply by 1, and you have the same abundance this year. So if you're above 1, you're increasing. If you're below 1, your population is going to be declining. And so you just multiply that number by what you were last year. And so these box plots are just representing some uncertainty in our estimates, but we can see that most of them are overlapping with that red line. And so this is suggesting that for many of these species, they're having pretty stable populations across the study period from the mid-90s. And then we can also kind of look how species composition is changing It's a lot to look at here, but essentially what we've found is that there's less scoter species now than there was at the beginning of the survey, and there's more, there's a higher composition of like mergansers and goldeneye species, but still largely dominated by scoter species. And then we can also look at sort of the community diversity across space as well, where the darker red spots are maybe where there's a higher diversity of sea duck species, and we might see along the coast line that there's higher species where those depths for foraging are kind of at those ideal levels. And then we also kind of see up in towards the Strait of Georgia, it's maybe a little bit darker, and the populations are maybe a little bit more diverse up there. Okay, so this takes us to, now the Puget Sound Seabird Survey and what we're doing with those folks. And it's kind of the next steps of what our work is going to be doing. And so a lot of you are probably familiar with the amount of community and citizen science that has been happening probably over the last decade, and we have a lot more information that researchers like myself can use in conjunction with some more traditional agency-based data. And so there's a lot of power to be able to use all of that data in conjunction. And so Toby reached out to us and we worked together to get this sea grant with the idea of bringing together what DFW is doing with what the Puget Sound Seabird Survey are doing. Toby gave a great overview of the program. And so again, these sites are across the Puget Sound and really stretches all the way out into the sort of the opening of the Salish Sea at the Pacific Ocean. There's like over 100 sites, and it's growing as Toby is getting more sites. But looking at the data, we kind of see similar trends between what the agency data is saying and the Puget Sound Seabird is saying. And you might think, oh, great, that they're agreeing. But then you have to think, okay, well, what's the benefit of bringing these sort of data sets together if they're kind of saying the same thing? Why do we need one, and why do we need to put them together? Or why do we need the other one? And so the great thing about the Puget Sound Seabird Survey is that it's happening over 7 months, which encapsulates the entire overwintering period of these seabird species. And it allows us to look at changes in seasonality, which is really a limitation of the agency-based data that's actually only happening at a very short period in their overwintering distribution. Let's see if that, there it goes. So only happening in December and January, right? And so we can't really look at seasonality or changes in the overwintering distribution of seabirds unless we bring in the Puget Sound Bird Observatory. And what the agency data provides is that it's, at sea, and they're going over areas that, people who are doing surveys along the coast aren't able to, see out that far, right? And so kind of using the strengths to help improve our ability to estimate what's going on and what's potentially driving distributions, and are they changing within the overwintering period. And so kind of getting back to thinking about, this survey and what are the issues with counting and observation air. So in the Seabird survey, we're doing point counts. We're looking out to 300 meters and we're trying to enumerate the number of birds and the IDs of those birds, right? And so here you might see, oh, I have 3 surf scoters, right? And so you count those birds and you've ID'd it. But if you look closely, you actually see that there was really 1 white-winged scoter mixed there with the surf scoters. And so this sort of concept of misidentification is also occurring in this data, or can occur, right? And so as Toby mentioned, there's a lot of different ways to reduce that. They are trying to pair folks that are really experts with people who are more novice. You're able to use resources like the other observers who are there, but also other information like a field guide or something along those lines to be able to reduce your chances of making an error. But then there's also sort of model-based methods to help correct for species myths ID, which I'm not going to get into, but there's things like that that can improve observation errors that happens out in the field. And so sort of similar again to the aerial surveys, we have this idea of imperfect detection, or thinking of it as counting errors where you're missing individuals, right? You've counted those 3 species, but there's actually a 4th bird there that we totally missed. And so we undercounted the total number of birds that were there. And we can see here that there was a rhinoceros auklet who happened to be hanging out, right? And so we, this was even a combination of not just miss ID, but also we just totally missed this individual, right? And so this is when this concept of distance sampling, if you're familiar with the Seabird survey, why you do the distance sampling, why you do the measurements, right? And so there's this really great relationship between distance and your ability to detect something, right? And it's just as simple as things that are closer to you, they're closer, so they're easier to see. Things that are further away, harder to see, easier to miss, right? And so we can use that sort of relationship to account for sort of the fall off in your ability to accurately detect everything that's there. And so this is sort of a function that exists that, you have this sort of detection probability. So right at 0, when you're standing right there, you should be able to detect everything at your feet with 100%. But as things start to move off into, if these were meters or feet, as things get further away, right, your ability to detect things starts to fall. And so we can use all those recorded distances to essentially build this function. And we can look at the area under the curve for those of you who remember your calculus. And we can get this detection probability, which then kind of gives us information of how many birds were missing during this survey. And so, we have these different ways of improving our counts, but another thing that we can do is building a model that then integrates both of these data together. And the way that happens is that we share this abundance. So back to that one where I'm showing, the abundance and the different demographic rates like births, deaths, and all those parameters, we can share those between these 2 different data sets. And it allows us to have better estimates of abundance and allows us to do cool things like look at seasonality or things that are further offshore. And then we can try to map those again to the different sort of disturbances that we might think or hypothesize are, changing the trends or the distribution over space. With the overall goal again of, looking at these different species, how they're doing over time, and thinking about sort of the marine ecosystem and potential recovery of Puget Sound and stuff. And so we're still in the process of, integrating these data sets together and exploring some of these relationships with the environment and with humans. So that's all I have for you this evening. I'm happy to take questions. So thank you.
Excellent. Maybe Matt, you can close your presentation, and Toby is about to have to be a parent, I think. So maybe we can start with Toby questions, and all of you feel free to type questions into the chat. And if you'd rather, you can unmute and turn your camera on. I think there must be, I don't know, 10, 15 surveyors out there from the PSSS, and we all know we do this thing where we do measurements and take bearings. And Toby, could you give a verbal short description of what that becomes?
Could you ask that question again? Sorry.
It's the one that Karen first put in about the bearing and distance, how that is translated ultimately into scientific data.
Well, I think you should, I'm going to pass it on to the other guy because it's called distance sampling, and he's actually worked with distance sampling data. So I'm going to hand it over to Matt.
Sure. Yeah, so essentially the reason we take a bearing in addition to taking a distance, right, is that we really need these perpendicular distances rather than sort of your straight eye line to it. So we use those bearings to kind of help us then get those straight line distances. And then, there's a couple of metrics that you take with, how long your arm is and some other stuff like that. And essentially, we just convert all that information into a distance from you to the individual. So all those measurements just go into getting that distance as accurate as we can, and then we build that relationship between distance and sort of our ability to detect individuals.
So Matt, as I understand it, I asked the question, and I think you actually answered it in your presentation, because what I thought I heard you saying earlier was it helps you decide how accurately we were able to identify that bird. Because if the distance and the bearing, if the distance is all the way out to the end of our 300 meters at an angle for us, maybe over by the barrier, maybe our identification is going to be less accurate. Is that what you were saying?
Yeah, so it's just that simple relationship. As the distance increases, right? It's just harder. It's harder for the human eye to see things, and so your ability to accurately count birds decays over distance. Similar to the idea of your hearing decays over distance, right? And it's hard to hear birds as well that are further away. So there's the limitation of the human senses.
May I insert a little sliver? I mean, what Karen and Matt just said is absolutely right. Our accuracy declines, but the reason that a non-calculus person sees this is that we're the 270 degree, we're taking little cookie bites along the edge of the Puget Sound, and the assumption on your part is that these birds are distributed equally along all the surface of the water. Is that not right? And so you're calculating an abundance based on our taking these little cookie bites along the edge.
Yeah, so the assumption is that within that 300 meters that there's the sort of the distribution of birds is uniform, but our model actually allows for us to look at changes in distribution over space. And so just because you're seeing, if you're where I'm at in Squim, if you're seeing that many birds, it might not be the same if you're up in Bellingham Bay, right? And so we can then tie those differences in the counts to different explanations that we're hypothesizing, whether it be the differences in prey or whatever.
Thank you. There is a question that relates somewhat to these methodologies also. A question earlier, what will you do with numbers of birds that are detected flying over the region? How will that interact with the other data? Did that make sense?
So I always ask folks to, focus on the birds on the water, because those are the ones we're getting, excuse me, a distance to and the bearing to, because they're not flying. It's very hard to get a distance and a bearing on a bird that's moving. So we ask folks to focus on the birds that are on the water with wet bums. The birds that are flying, if you've got time and you've recorded all the birds in front of you on the water, then by all means make note of the birds that are flying as well. But yeah, your priority is birds on the water. But the birds that are flying, we can also use that for maybe there's a species that we don't record very often, and if you see it flying, then at least we can get like a point at that particular location. And also just for general abundance of birds, whether they're flying or sitting on the water, we can estimate that as well in a very kind of loose way.
While I try to voice this next one, please encourage you to put your questions in the chat or unmute and get ready to talk to us. The question about optics, for those of you who are not yet participants, we were asked in each survey to put down what power binoculars and what power of scope we're using. And I'm sure that a site that does not have adequate objective powers is going to be maybe factored in a little differently. But I don't think the quality of the optics is something that you can gather. Is there any comment about that? Gene was asking, what about the quality of the optics?
My only comment was to do with how, depending on what analysis we're doing and how strict the requirement for that analysis, we may filter the data based on what optics have been recorded at, for those particular surveys. So if people have just been using their eyes like this, we might filter that out. But people have been using, a 60 times scope, then we'll include that data because it's potentially going to be pretty high quality. So yes, that's why we record that information. And yeah, optics have definitely improved over the years, of course. So yeah, we just ask for that general information in terms of how powerful the optics are, and we kind of just leave it at that. And if there's some weird anomaly in the data, then we'll axe that data potentially. Yeah, Matt.
Yeah, I was just going to jump into, so there's some other sort of information like the sea state that is collected and basically things that are maybe influencing your ability to observe birds. And so in addition to potentially filtering, you can also, similar to how I was talking about differences in abundance over space, you can look at changes in detection probability over space and time, and you can account for some of that variation in your detection by building relationships between things like sea state, or what your magnification is on your scope or your binoculars. And so essentially we would have information then to predict out to detection probability for like, oh, if the sea state is this, if it's a 1, I know that my detection probability is going to be this because of that, versus oh, I'm using this magnification, I'm gonna have a better detection probability than if I was using a lower magnification. And we can build that into our statistical model and sort of capture that variation.
Matt, where's your site? I want to come work with you. I mean, you can walk the talk.
Yeah,
You can do the math.
yeah, I can do the math. My Seabird ID skills and even my counting skills are there's much to improve. But yeah, we have sites out in Squim, that's kind of on Klein Spit near
Very
there. Yeah.
Elaine,
I have one more question, Karen, excuse me. I have one more question that I'm going to present to you all, and it may be maybe the best question of all. Those of us who participate as laymen, laypersons, citizen, or community scientists could be criticized compared to studies that are done with rigorous protocol. And it's an eternal question. If you could have soldiers taught to do exactly the same thing at every time, at every point, that would be the ideal. Karen was asking, do you think the data will be challenged on the grounds that it's collected by citizen scientists? And to me, that answer is that dichotomy always exists. And when you write the paper, when you write the protocol, and when you analyze it, you put all the details you can in there. Comment?
Yeah, I have 2 comments. So the first thing that I think folks will find interesting is that figure I showed earlier on that was quiz respondents and observation error over time. So they sent that out to experts who do aerial surveys all the time, and they also sent it out to people who were non-experts. And so in that case, they found no difference between people who do it for a living versus people who don't. And so there's just a limitation of people's ability to count flock sizes. Now, species ID is a different sort of story where if you do it more, you're going to be better at it. But the counting thing, you can try to get better at it, but honestly, there's a certain limitation that humans have in being able to accurately count something that big quickly. And then the second thing, I would say that if there's thought that goes into any sort of monitoring program, regardless of if it's done by an agency, if it's done by a group of community members, if you're accounting for the certain errors that I'm talking about in your protocols, which for the Seabird survey, the distance sampling, and sort of the training on species ID that happens, if you're doing those things, your data should be, at a quality that, scientists are collecting as well. And so it really gets back to spending time developing those protocols, and thinking about what your questions are, what you're collecting the data for. But at the same time, as you move in time, going back and assessing those protocols and seeing if you need to update them. I know there's a lot of people who are hesitant because they want the data to be consistent, but people who are like me who do the back end, it's like I can account for changes in protocols. That's no problem. But if you improve your protocol, I can maybe even retroactively go back in time and account for the fact that you weren't doing distance sampling, right? You didn't have information on, undercounting. But if you're willing to continuously think critically about what you're trying to do and improve those monitoring protocols over time, I think any sort of person collecting data, it will be robust.
Perfect. I know that in some fields, if I may mention it, say in the medical field, they'll ask so-and-so to look at these photos and look so-and-so to look at these that's not in the medical field. And sometimes they will get it better because they're doing it more carefully.
Sure.
At
Right.
any rate,
Fatigue.
yes. Here's a question. Oh, no, it's one of the many thank you for a great presentations. If anyone else has a question, we'd love to hear you. You can just pipe right up. With that being where we are, I think, Matt, we cannot thank you enough. You've gone both ways for us, taught us a great deal about things. Thanks ever so much.